what I'm going to talk about represents a single example of a rare disease that fits the mold for LGS and where you're headed, and I think the end point will be the value of registries. We have 10 years now, and the registry is really paying off. So the story is a personal story, partly because I come to you as a parent and a scientist researcher. The story begins with my daughter, who was born in 1996 and had very undiagnosed seizure disorder, difficult to control seizures, multiple seizure types, developmental delay, characteristic EEG of Lennox-Gastaut, actually. I'm a scientist. I was doing population genetics and sequencing genomes, and at some point I just got fed up. When Shay was about 14 or 15, we started a whole genome sequencing project with Shay's mom, dad, and brother, and so we did a quartet, and she became the first patient with SCN8A related epilepsy. First known patient, I should say. It's humbling to think that people throughout the eons have had SCN8A epilepsy, but my daughter became the first known patient. All I knew about SCN8A was through Shay, my daughter. But I want to say that the other story here is that we've been able to make very, very rapid progress since 2012 when we published the gene. And as of 2024, we have clinical trials, a couple of different small molecules in trial. We got mouse models with ASOs, and we have caregiver and parent-supported groups that have come forward to really help promote things. And I think the story we have here is really one of just passionate parents who have found the right researchers, and the researchers have found each other, and passionate clinicians like Scott, who have been very involved with us. So I think our success is due to many factors, but I think one of them is this registry. So in 2012 when we published, all I knew was Shay was the patient with this disease. But I started to get emails about 2013, 2014 saying others had been diagnosed. And I said, oh, be careful what you wish for. I always wondered if there was anyone else like Shay. And so I started an online registry, and I designed the questionnaire to be very detailed, but very much following what I knew about Shay. It's an extensive survey tailored to the wide spectrum of characteristics because we've iterated the survey a few times as we've learned more and more about the spectrum. And we've collected caregiver-reported data with the central goal to advance research and improve quality of life. We see the registry as central to helping clinicians and researchers alike. Pharma have been aided by it for creating a natural history. And of course, the caregivers are the central players in this, and that's why the title is Caregivers to the Rescue, because it's their data that have been entered into the registry. We have several goals listed here. We found genotype-phenotype associations. We've identified patient subgroups through machine learning approaches. We're aiding in clinical standard of care, aiding in the design of clinical trials, and we're integrating our registry data with medical records. Just a few words about the registry. There are now something like 17 different modules. This shows you something like 12 of them. Everything from birth and pregnancy to comorbidities, puberty, growth, genetics, medications, developmental delays, tests and procedures. We can upload EEGs, MRIs, and all of that. And we're international. We have patients all over the world. We have network meetings with patients regularly to talk to them about their concerns. So I'm very plugged into the international community. And fortunately, after 10 years, we have over 500 registry participants. Just a bit about the demographics. You can see the age distribution on the left. It's very much skewed towards younger ages, like 6 to 9 years old, with a mode there. But we have patients going all the way up to 60 years old. I was listening to the discussion about transition from peds to adult. We have a lot of patients who are about to make that happen, make that transition. So I was very interested to hear what you all had to say about that. We have about a 50-50 sex split, and about 90% of our patients have seizures. But that's because we not only have gain-of-function variants, we have loss-of-function variants. So we have a very broad spectrum in this disease, ranging from no epilepsy shown down here at the bottom, with just behavioral movement disorders to benign familial epilepsies to mild to moderate and severe DEE. The gain-of-function group, almost all of them have seizures of various sorts. The loss-of-function is about 54% have seizures. They're more like generalized or absence seizures. You can see sort of a word cloud in the letter L over here for loss of function, and a word cloud over here in the G for gain of function, with many comorbidities. Like I said, the family organizations have really helped promote everything. I started my own foundation called the Shama Research Foundation, and joined forces with Wishes for Elliot, a mom and a grandmom who started their own foundation. We formed the International SCN Alliance, which really does put families at the center and really helps promote the registry. And as I said, we really interact closely with clinicians, industry, researchers. This has been a big help. For example, we had a survey that, we had a workshop last summer that was essentially a research roadmap, not unlike what you all are trying to do here. We went through it last year, and before we had the meeting, we designed surveys for families, for doctors, for scientists. We tried to harmonize the questions in a way to see how families' needs were supported by clinical clinicians and scientists, and how scientists and clinicians were corresponding with each other. This is just showing you one chart of the caregivers' answers to the comorbidities and top 5 quality of life challenges. You can see seizures do rank very high, but so does cognition and speech, motor-related sleep, behavior. We have all of it in this one gene with all of these different features, just like in LGS and so many other rare epilepsies. The registry initially was very helpful. We did supervised learning, and we were able to find statistical support for 3 different gain-of-function subgroups within the patient cohort, and loss of function as well. And last year there was a publication in Epilepsia, 2 papers published. Elaine and Scott were a big part of this. We had the first consensus on diagnosis and phenotypes, treatment, prognosis, and comorbidities. So we have our guidelines. Unfortunately, we didn't find a lot of consensus. We had 30-some clinicians around the world in a modified Delphi process, and we agreed on 5 phenotypes. We also agreed that the gain-of-function subgroups do better on oxcarbazepine and carbamazepine, 2 sodium channel blockers, as first-line treatments. However, we were not able to reach high consensus on many of the other things we were looking for in our guidelines. So this was a great start, and I think it will be followed up with more work. Now I want to talk briefly about the approach to do data analysis with the registry data that we have. We started out with input on seizure type, seizure onset, developmental profiles, genotype, etc. Our first publication was on distinguishing loss of function and gain of function patients using machine learning. We did not, we based it on a truth set that was based on in vitro biophysical properties of cells in culture, and looked at the phenotypes of patients so that we could make a machine learning assessment of whether patients were loss of function or gain of function. The most important first step in prescribing medications. You want to generally avoid sodium channel blockers for loss of function, and they're very helpful for gain of function. The next series of studies we did were basically identifying patient subgroups and looking at the prognosis of those different subgroups. And now we're on to using the data to inform clinical management and trying to identify the best drugs for these different subgroups. I'll tell you a bit about the subgroups here. We're kind of going backwards in the history of DEEs. We're deconstructing DEEs into DEEs as being a combination of both developmental and epileptic encephalopathies, but we do think there's separate entities that exist. When we use unsupervised machine learning and let the data tell us what was going on with respect to patient subgroups, it revealed something we weren't expecting, and that was there's some patients who have developmental impairments primarily with seizures, and patients with epileptic encephalopathy, so that when their seizures are controlled, their developmental skill acquisition improves. However, those with the developmental impairments and their seizures are controlled do not do as well in accelerating their developmental trajectories, and then developmental and epileptic encephalopathy were the more severe patients in both groups. These are not black and white groups. We call it a partially ordered landscape. And you can see on the left the relative frequency or proportion of these different patients, including both loss of function groups, the DEEs, about 50%. It's also very interesting to us that now we've been looking at developmental acquisition, and these subgroups differ fairly dramatically in their developmental profiles as they age from 3 to 5 to 8 to 10 years old. You can see the developmental encephalopathies pretty much flatline, no matter how well their seizures do, where the epileptic encephalopathies continue to make progress, and that's generally due to seizure control. The DEEs are in the more unfortunate situation, and then you see the loss of function group. And so loss of function does share a lot of properties with gain of function. So having said all that, as a quick summary of how far we've come, there are many gaps that remain. The disease mechanisms are not fully understood. We lack treatments for non-seizure phenotypes and comorbidities. We lack strategies to deal with a high premature death rate. We lack biomarkers to track disease progression and treatment response. I know there's been a lot of discussion today about biomarkers. We fully concur how important that is. And what I'm just going to focus on for the rest of the few minutes I have is how we can use current approaches that we've been talking about from EMR and from the registry to improve our ability to choose medications that are likely to be beneficial and determine which ones are detrimental to try and avoid those, given our very low rate of seizure freedom. Like you've been mentioning with Lennox-Gastaut, we have about 85% with intractable seizures. So we had a research roadmap meeting last summer in Boston, just very much like this meeting. We had we had these surveys with 175 caregivers, filled them out, 46 clinicians, 23 scientists. We tried to identify gaps in our knowledge with respect to this disease and which research priorities would be the best to promote to fill these gaps. I'm not going to talk about all, but we divided up into 5 working groups, sort of like what you guys have been doing here. The first working group was transformational therapeutics, which was gene therapies and small molecules. The second was non-seizure outcomes. The fourth was biomarkers, and the fifth was whole body, whole brain, whole body, we called it, to show how important the whole body is, an epileptic body, not just an epileptic brain. And what I'm going to talk to you about now is the part of this roadmap that I'm working on is to improve therapeutics on a shorter time scale than realistic for. We're going to wait years for gene therapies, we're going to wait years for some new molecules. So what can we do today to improve current therapeutics, and do registry and EMR provide the necessary fine-scale data to determine the efficacy of given drugs? So we took electronic medical record from using the citizen data for SCN8A, and we wrote code to produce these longitudinal profiles for every single patient. There were over 80 patients that participated. We got the data, and then we wrote this code that shows you on all these different lines the medications they tried in order of addition, when they weaned them, and the next lines are adverse events. Over the course, you can see the very bottom is the age of the patient in months. This is 60 months for this patient. And all of the clinical office visits were shown down at the bottom in these green triangles. We also show the different seizure types, the intensity of the seizure types as reported in the clinical appointments. This particular patient had tonic seizures, focal seizures, bilateral tonic-clonic seizures. We have EEG reports showing when the EEGs were normal or abnormal. We looked at status events, we looked at milestones that were acquired. We showed registry data for skills that were acquired in the first 50 months of life in fine motor, gross motor, language, and social motor. And we have a heat map for how medicines did in terms of improving the outcome for the different seizure types in this particular patient. We have this for 80 patients, and this is our way of saying we are now going to try and correlate medications that were added, weaned, when there was periods of seizure burden reduction, and what medications were used in those so-called gaps. And I'm going to show you some results that we have. In looking at the period of time when there was reduced seizure burden, it's the best we can do given the complex nature for all these patients on many drugs, polypharmacy, rules of the day. But this patient, when they started valproate, rufinamide, they were on gabapentin for the whole course, but these 2 drugs seem to make a difference. And they show up here in the lighter blue heat map as drugs that seem to make an improvement. The drugs that, if we look at all the patients, you look at those that are on monotherapy, dual therapy, and polytherapy in these different bar charts down here, you can see the sodium channel blockers, the GABAergic drugs, calcium channel, levetiracetam, multi-mode of action drugs, and other shown as drugs that were given during those periods of reduced seizure freedom. The monotherapy drugs, which I thought were most informative, for patients on monotherapy and those reduced periods of seizure freedom, it turned out to be all sodium channel blockers, oxycarb, lamotrigine, phenytoin, lacosamide, and carbamazepine. So that's something we can, squeezing some water out of the rock from the electronic medical records. What we've also done is use the registry data to select effective medications using a machine learning approach, incorporating a metric for improved seizure outcomes that involves caregiver assessment. So it's kind of like a clinical outcome assessment that involves the caregiver input, which we think is so important and is really very consistent with FDA guidelines going forward. So we constructed a neural network that recommends ASMs based on caregiver-centric composite measure, incorporating changes in seizure control, alertness, and side effects. And we compared these recommendations of the algorithm with the preferences of clinician experts in the clinician game to see how well clinicians and this algorithm matched up. And so what we found, here's a picture of the very complex medicinal landscape that the patients are on right now, with a number on monotherapy, dual therapy, and polytherapy. As I described earlier, this is without the algorithm. But when we use the algorithm to try and identify those drugs, those medicines that were making a difference with regard to caregiver input, we were able to identify about beneficial drugs in about 65% of cases of these runs, these computational runs, while almost never recommending a detrimental ASM. So regardless of being able to find the right good drug, the algorithm really helps in dissuading you from using a detrimental drug. And this was despite the very complex medicinal landscape of polypharmacy and frequent suboptimal treatment responses. And so what we found was we kind of expanded what the consensus was able to do in recommending oxycarb and carbamazepine. Now we also find that the algorithm prefers lacosamide, lamotrigine, and phenytoin. And these are the same drugs I just showed you in the previous slide that the electronic medical record gap analysis was pointing to. We also see that there is a GABAergic drug, clonazepam, that looks like it's one of the more beneficial drugs, and there are several drugs that are recommended as detrimental, especially levetiracetam, which is one of the first prescribed drugs in this epilepsy and many others, as well as phenobarbital. So we've reduced the very complex medicinal landscape from over 15 to 18 drugs that were used by people participating in the registry down to 6 that we would recommend as best first-line medications. And as I said, the approach we're taking is consistent with FDA guidance. I just want to point out that the problem remains in terms of estimating on a granular basis accurate seizure frequencies. Because the EMR, those patients only met on average with their doctors about every 2 or 3 months. The clinicians would glean information or gather information on how the patients were doing for that period of time. It's not very granular. Even the registry, we don't have granular data collection. We have an annual update, and we've been able to estimate from annual data that there's about 8 seizures per month as a median. We don't have any good estimates from the EMRs of how frequent seizures are. And so we think this is a really important next step, is to try and get granular seizure frequency change data. And so we've initiated a pilot prospective study to collect high-quality multi-source data, including daily seizure and medication entries from caregivers, and incorporate that with relevant medical notes from the EMR based on office visits and quarterly survey data from the registry on development, acquisition, and quality of life assessments. And we think by integrating all of this together, we should be able to come up with much better correlation analyses. This is an example of the electronic diary app we're going to use, and it shows you just a single patient with the medications in dots and lines and seizure frequencies in bars. And the basic prospective study design is we have a 3-month setup period where we recruited 50 patients. We give them training. We have a 25-patient cohort now that are very compliant users. Now they're entering the 9-month follow-up study, so we'll have 1 year of
daily seizure and medication data. The app also collects mood and quality of sleep. And we're going to integrate this with clinical data from EMR, quarterly survey data from RedCat, our own survey, and daily seizure medication data for our studies on correlation and machine learning and prospective model building. As I said, this does fit nicely with draft guidance from the FDA. Clinicians and caregivers have different approaches to estimating how well these medications are working. And just to summarize, I'll say that this also comes from the FDA. The advantage of the registry and the work that we've done is right here in the slide that the FDA's put out with the patient caregiver perspectives included and natural history of disease and the condition that we're building now. We've identified patient subpopulations and how we are involving medical records as well as patient caregiver determined feedback. So I will end it there. Hopefully I finished in time and thank the people that have worked so closely with me to do the data analysis. We were funded by two biotech companies, Praxis and Neuroprint, as well as the Shayla Research Foundation. And the folks, including the International State Alliance, Women, Gabby, and Jada, and my research group, all contributed significantly